Okay. So, do you want to start?
I don't know how to start.
Just saying a name. Okay, I'll start. Hi, my name is Harleen Bailey.
And I'm Farwa Jaffrey.
So, we are doing our presentation on CRISPR and how is it applied to technology or biotechnology. So, what is CRISPR? CRISPR stands for clustered regularly interspersed small repeats. In smaller words, it can be used to cut and edit genes. CRISPR technology is a simple yet powerful tool for editing genomes. It allows researchers to easily alter DNA sequences and modify genes functions. Its many potential applications include correcting genetic defects, treating and preventing the spread of diseases, and improving crops. The application of CRISPR. Researchers studies CRISPR has adapted for use as tools for genetic modifications of the target host genomes. CRISPR has currently become a popular genome editing tool due to its simplicity and versatility. To simply describe this technology, CRISPR means gene mutation or gene editing. Genome editing technology enables scientists to make changes to DNA, leading to changes in physical traits like eye colors and disease risk.
Okay, so an interesting fact about CRISPR technology is that it's adapted from the natural defense mechanisms of bacteria and archaea. These organisms use CRISPR-derived RNA and various Cas proteins, including Cas9, which is why it's called CRISPR-Cas9, to foil attacks by viruses and other foreign bodies. They they do so primarily by chopping up and destroying the DNA of a foreign invader. When these components are transferred into other more complex organisms, it allows for the manipulation of genes or editing. CRISPR. So it's had a very long history. The mechanism was first published in 1987 at Osaka University. The classification of CRISPR-Cas system was proposed in 2011. The first commercialization of the CRISPR-Cas9 technology was in 2012, and CRISPR-Cas is first used in human genome editing in 2013. CRISPRs are basically specialized stretches of DNA, and Cas9 is an enzyme that cuts the DNA apart.
Components of CRISPR. How does it work? Editing the genomes of yeast, bacteria, mice, and other organisms that scientists commonly study has led to countless discoveries about how the genome is connected to physical traits. So, scientists cut DNA with CRISPR to make gene changes or mutation. By comparing cells with and without the mutation, scientists can sometimes figure out what a protein's normal role is, or a new mutation may help them understand genetic diseases. So, the process of CRISPR. So, the first step is adaptation, where DNA forms an invading virus is produced into a short segments that are inserted into the CRISPR sequence as new shapers. And the step two is production of CRISPR RNA, where the CRISPR repeats and spaces in the bacteria DNA undergoes transcription, the process of copying DNA into RNA. And lastly, step three is targeting. CRISPR RNA guides bacteria molecular machinery to destroy the viral materials. Because CRISPR RNA sequences are copied from the viral DNA sequences required during adaptation.
So bioethics, should CRISPR be used to modify humans? There are a lot of arguments about this, and Um, on the next slide, it shows that the arguments. So the first argument that people make are the safety concerns. We know that the technology is improving, but scientists will need to do many more studies before it can be proven safe to use on humans. And number two, technology is improving. So people argue that if it's improving, then we should be able to help others and be safe because we have the ability to um, make this technology work. And number three, uneven distribution. People argue that if we do allow germline editing some way that it could lead to the creation of designer babies whose DNA has been carefully selected to enhance their appearance, intelligence, or other traits that are not medically necessary. So these enhancements may also be unevenly distributed among the population, and it would lead to a society of genetic haves and have-nots. Number four, disease prevention. So if we have the technology to prevent diseases that are fatal, why shouldn't we use it? And those are like the basic arguments involving CRISPR.
How CRISPR came to be. So, CRISPR is relatively a new technology, but it has covered a lot of ground in just a few years. Mexica was the first researcher to characterize what is now called as CRISPR reported in 1993. He worked on this throughout the 1990s and in 2000. He recognized that what has had reported has discredited repeat sequences actually shared a common set of features. In 2005, he reported that these sequences matched SNPs from the genomes of bacteriophage. This finding led him to hypothesis correctly that CRISPR is an adaptive immune system. So, why are scientists so excited about CRISPR technology? Has scientists had the tool of editing human genomes before the discovery of the CRISPR? However, these tools were expensive and labor intensive and sometimes did not work for particular DNA sequences. Because of the CRISPR system came from bacteria, it is it is cheap and easy to produce the necessary protein. Additionally, the CRISPR system can easily be targeted to any DNA sequences. Lastly, it seems to be fairly specific, meaning that it should only target the specific DNA sequence that scientists program it to target.
So recent research about CRISPR. In April 2017, a team of researchers released research in the journal Science that they have programmed a CRISPR molecule to find strains of viruses such as Zika in blood serum, urine, and saliva. On August 2nd, 2017, scientists revealed in the journal Nature that they had removed a heart disease defect in an embryo successfully using CRISPR. On January 2nd, 2018, researchers announced that be they might be able to stop fungi and other problems that threaten chocolate production using CRISPR to make the plants more resistant to disease. On April 16, 2018, researchers upgraded CRISPR to edit thousands of genes at once, according to research published by the journal BioNews. And the most recent research involving CRISPR technology, there's an article published on July 10th of this year, which was only a few days ago. The approaches of genetic manipulation in streptomyces were discussed. And what those are basically a type of bacteria, and they have like 500 different species. There has been a toolkit developed by scientists that include different variations of the CRISPR technology systems that are specifically used for those bacteria. And careers involving CRISPR. So the first one is a senior scientist and medical director and project manager and translational pharmacology project manager. And what they do is support translational pharmacology in the development and execution of biomarker plans and toxicology plans for drug development or research programs. And for all of these jobs, you either need a bachelor's or a master's and at least 3 years of experience.
Thank you. Mm.